So thank you all for coming today. I'm really excited to share my research with you all over the evolution and biogeography of the genus Triodanis. So my dissertation broadly covers four topics over three chapters using the wonderful Triodanis here to explore different ecological and evolutionary patterns. And I did this through a couple different means, primarily by exploring and understanding the evolutionary history through a genus-wide, um, sorry, got choked up there, phylogenetic study. I followed that up with a morphological study using both field and herbarium data to see how those relationships match or don't match the phylogenetic trends that I was seeing. I then looked at the species level ecological niche to see how these individuals were using the landscape. And then lastly, I wanted to understand population connectivity through the use of paleoclimates. So understanding evolutionary and ecological relationships is really critical to kind of fully understanding the processes of our study system, especially in terms of things like hybridization, responses to climate change, trait evolution, and then kind of putting all this together to create well-rounded and useful conservation plans. And understanding all of this kind of provides a backbone for your research so you can use that context when exploring all of these other topics. And my research kind of briefly touches on all of these topics here and then tries to use that to implement them in kind of a pragmatic sense. So first, I'll start with a little background on Triodanis here. It is within the Campanulaceae family, which is the bellflower family. This family is huge. It's got approximately 84 genera and 2,400 species, and it is located across 6 of the continents. So there is a great diversity in distribution and morphology here. And there has been a lot of research because of this into the family. But because of its size, that leaves some of the lower level groups kind of less explored. So that is where I came in. And I focused in on Triodanis, as most people in the Weber lab do. And I use that to answer all my questions here. Triodanis currently has 7 described species, and the range of those varies pretty widely, as you can see here, from across much of North and South America, all the way down to these narrowly endemic 2 here that you see that are just located in like South Central Texas. Most of these species are weedy and relatively common, which is awesome for me because that meant that it was relatively easy to find on the landscape, especially using things like iNaturalist to kind of plan that field work and make it easier on myself. So first, I want to present the newest comprehensive phylogeny for the genus. And one of our big questions was the relationship between Foliata and Biflora. There's been a lot of discourse about whether or not Biflora is its own species or if it is considered some sort of subspecies of Foliata. And Biflora was first described all the way back in 1779. And then it wasn't until Bradley here in 1975 kind of revisited those relationships. And he used a really comprehensive experimental morphology and hybridization study. And he concluded that he thought Biflora and Foliata kind of represented 2 populations that are incompletely separated reproductively. And then he classified it as a variety, Biflora as a variety of Foliata. And then Lammers followed this up in 2006 and kind of revisited the study to clean up some of the species name in Campanulaceae since it is so huge. And he agreed with what Bradley had seen here, but changed Biflora to a subspecies of Foliata. So I understand why they did this. Biflora's range is almost completely encompassed within Foliata's range, so almost everywhere Biflora occurs you have the potential to find Perfoliata. And it is also well known throughout the literature that these species, along with other species of Triodanis, hybridize readily, and they do it quite often. That said, previous work in the Weber lab by Keegan here found that morphologically, these two species are distinct even when co-occurring. So he collected in populations where Perfoliata and Biflora were touching. He collected 5 transects and measured 23 traits on approximately 200 individuals. And you can't totally see the separation in 2D here, but when you bring in that third dimension, Biflora here and Perfoliata almost form completely separate morphological groups, which is important to note because, like I said, these are all at populations where they're co-occurring, so there is the potential for hybridization. And then previous work by Crowell et al., he was looking at the phylogenetic relationships within Triodanis and sampling across 17 individuals and 6 gene regions, created this maximum likelihood tree you can see here. And while he got good resolution between Triodanis and the then sister group Lagusia, when you look within Triodanis, you can see that the relationships are totally clear. For instance, Perfoliata here is kind of jumping around as well as Coloradoensis. So while this was an important first step, it was clear that further sampling would be required to elucidate these relationships. So that's where I came in. My first goal was to increase the sampling that we had. So I spent 3 field seasons going across as much of their range, collecting from as many populations as I could, especially populations where species were co-occurring. I did make note not to collect intermediate phenotypes because for my study, I was focused on broad phylogenetic relationships, and we understand that these other processes are going on, but we can only answer so many questions at once. So I had to start somewhere, and somewhere was here. So at approximately 40 of my field sites, there was at least 2, or sometimes I think up to 5 Triodanis species present. So we made sure to collect from all and sequence and try to include as many of those as we could. And then my final data set ended with 125 Triodanis individuals supplemented with 14 herbarium specimens from the Missouri Botanical Garden. So next, I wanted to work to expand the genetic sampling, and I did this using a contemporary probe set called Angiosperm 353, which is broadly applicable across all angiosperms. This probe set targets 353 low-copy protein-coding nuclear loci that are present in most angiosperms. So this is really helpful for a number of reasons. It makes your work kind of comparable across studies, whether or not you collected that genetic data. It also allows for phylogenetic analysis without a species-specific probe set, which most people don't have access to. I definitely didn't. And it's also been proven really useful in uncovering both deep and shallow level phylogenetic relationships, especially when using herbarium specimens. What's important is I also included herbaria in my study here. So of these 353 nuclear genes, I was able to recover 168 in my final data set. And that brings me to my beautiful tree here. First, I would like to note that bootstrap values below 90 have been removed. So I know it looks like I have no support values, but my tree actually was just really well supported, which is awesome. So again, this represents 125 Triodanis I collected in the field, as well as 41 outgroup representatives. So just broadly here, first we can see most species of Triodanis form monophyletic clades that are well supported, which is really awesome for me. But I wanted to kind of collapse those monophyletic groups so we can zoom in and get a better look at what's going on here. So let me walk you through this tree, starting down here at Ligusia. So, shoot, I guess 5 years ago now when I started, Ligusia was described as the sister group to Triodanis. So my sampling was really focused on explaining this relationship between Ligusia and Triodanis. However, partway through my research, the groups were reclassified. So the sister group to Triodanis was changed to these three species you see here. So unfortunately, my sampling wasn't really targeted to explain the relationship between these and Triodanis. However, I did do a good job showing that Ligusia, in theory, would have been the sister without these here. So then as we're moving up, as you can see, we have Lamprosperma, Coloradoensis, Leptocarpa, Bolzingeri all collapse into monophyletic clades, as well as Biflora up here, which was really important because, like I mentioned, I was interested in explaining the relationship between Biflora and Foliata. However, I would like to note that these two rascals down here kind of messed up the monophyly of the Biflora foliata clades up here. And these two, represented by the circle, were both collected in the field by me. And when we were in the field, we used morphology to identify them to Perfoliata. And I think it is important to note that they occurred at a field site with 4 other species present. So based off of this, I think that there's obviously something weird happening here. So whether or not these are hybrids or some sort of other evolutionary path, I can't quite say right now. But the New Big Lab has been working to create PCR fragment analysis using DNA markers, which they've been able to do for 4 of the species. So I hope to use this as one means to kind of explain what's going on here. Okay, so that seemed like a lot, but really that was the easy stuff. So the last species I have up here that I haven't mentioned is Texana. And first I'd like to say that Texana and Perfoliata morphologically are almost identical. The key really only states hair and seed morphology as the difference. It basically said Texana is very hairy, Foliata is not, and that Texana has what's called quadrangular seeds, which are kind of like a diamond shape. So we were going off of this morphology. However, when we got back to the lab, we looked at seeds from Foliata across this range, both within Texas, where Texana is, and outside of Texas. And we found that Perfoliata has the same seed morphology across its range. So clearly that alone is not enough to distinguish the species. And I say all this to point out these 4 triangles you see up here, which are all individuals that in the field we identified using morphology to Texana. And they also all co-occurred with at least Perfoliata, if not other species at those sites. So at this point, I think we can fairly confidently say that these should be actually ID'd as Perfoliata. I'd like to use those DNA markers to kind of verify what we think here. And if that's the case, then I could basically collapse Perfoliata as monophyletic. So that brings me down to these 3 individuals. And the 2 with the asterisk here actually represent the type specimen, which was at the Missouri Botanical Garden that they allowed me to sample, which was really, really awesome for this study. For one, you can see here that the herbarium samples are totally separate from anything we collected in the field. And I'm sure many of you are wondering why I have the type on here twice. That's because we sampled it, and then the closer we looked, we said, wait a sec, that's 2 on one sheet, which is not generally how that should go. But while we were there, we looked a little closer at the morphology. And I know it's kind of hard for you all to probably see back there, but this stem is connected to the larger individual. And then there's a different root system here that when we looked closer, it wasn't like, there was no signs of tearing, like it fell off of the larger specimen. It was just two different individuals. So then we started kind of pulling back the leaves to look at the seed capsules. And we found that while this larger individual, represented by the two asterisks here, matched the original species description, the morphological description, this smaller individual actually didn't match their description. The capsule morphology was a little weird, and it kind of reminded me more of different species, which I think maybe Lamprosperma or some sign of a hybrid. So the fact that they are not together perfectly in my tree kind of, it makes sense to me. I'm not super concerned. I'd also like to note that these were collected back in 1917, and then this other herbarium specimen was back from 1966. So there are a couple reasons for these relationships. We're not sure if maybe now just Texana is exceedingly rare, maybe it's extirpated, or perhaps it's hybridized with other species enough that it's hard to find just pure Texana in the field. So I followed this up with an astral tree, which has slightly different assumptions than IQ tree. This works under the coalescence-based approach versus the concatenated approach that IQ tree takes. And and just to note, this uses posterior probabilities. So values above 0.9 are considered like really good support. So those have been removed. And then 0.89 to 7.1 is kind of, they're pretty good values. So those would be the exclamation points here. And then values below that are kind of poor support. But generally across my astral tree here, the relationships were the same as my IQ tree, which is what you expect, or what you hope to get your relationships be mostly the same with maybe a few minor differences. And those two main differences here are that those, what we ID'd to Perfoliata, are now hanging out with Holzingeri, which was also at the site where we collected. So we definitely need to explore these two further to see what's going on there. And then that specimen from the type that I thought didn't match the morphological description for Texana is now up here on its own. So again, I'm very interested in what species that might actually represent. And then if we go down to the sister group here, those two other Texana from the herbarium are kind of pairing all together. So now this leads us to wonder, should the sister group kind of be folded into Triodanis? Should Texana be folded into the sister group? Or what this relationship is here? But unfortunately, like I mentioned, my sampling was focused on the Legucia Triodanis relationship. So this will definitely require further sampling to kind of tease apart. So we have those outstanding questions surrounding primarily Texana. But as I mentioned, the new big lab is working on those PCA fragment analysis and hopefully can get the remaining species completed. And we actually just recently submitted that methods paper for publication. So that is very exciting. So next, I looked at the morphology and how it related to these phylogenetic groups. And I did this using primarily field collections. Unfortunately, though, this sampling was destructive, which meant that I couldn't use the same individuals that were represented in my tree, but I used individuals from the same sites that were in my tree. So I was able to represent 22 of the 41 field sites that were in my phylogenetic trees, and 1/3 of those had multiple species. So again, that's really important as we know that they are hybridizing. And if there were multiple mature species present at a site, I was sure to measure from all of them. So with the help of James down here, who I actually could not have finished this part of the project without, I measured, or we rather, measured 28 morphological traits on 318 different individuals. And I know it might look like all of these species are overlapping here, which is not what we want. But these are following the exact trend that I would kind of assume would that we would see here. So at the bottom, we have Coloradoensis and Leptocarpa, which, as you can see here, are very similar morphologically. They're very like slender and linear, both in this seed capsule here and the leaves. So I would hope that the individuals we measured did kind of overlap like this. And then at the top, we basically have the rest of the species all overlapping. And again, this is not really surprising to me because even as I put these pictures on the slide, especially these two leaves, I immediately had to go check myself because I wasn't sure I could tell them apart. And then this overlaps too with this seed capsule morphology. So their overlap is kind of expected, but I think it is important to note that when we looked at the third dimension, despite this overlap, it did sort of start to pull apart and form distinct groups between the species. So next, I wanted to do a MANOVA to see if these groups were significant. And I think this illustrates really well what I was saying about Coloradoensis and Leptocarpa being wildly different from the other 5 species. So I ran a second one to kind of focus in on those 5 most morphologically similar. And here, first I'd like to note that Biflora and Foliata again form distinct groups, which is awesome and exactly what I would hope to happen. And also, I think it's important to note that this green represents Texana, but as we saw based off the tree, I find it hard to confidently say that all of these individuals are Texana. So if we kind of think of them as Perfoliata for a second, it almost makes more sense because it would kind of encapsulate all these purple X's you see and still be fairly distinct from Biflora. So then again, I just wanted to zoom in on kind of the most significant morphological traits that these analyses pulled out. And that would be leaf shape, which kind of all look the same from this, but I promise there are small differences. And then the other main trait was related to the breeding system, but that's kind of a whole other bag of worms. So I can talk about that more later, but definitely looking at the capsules here, it's really important to tell the species apart. And leaf trait and capsule morphology are basically the main things that the key list as identifiable. So we did a good job there. Okay, so next I wanted to look at their ecological niche for each species. I would like to note that this was done at 1 kilometer, and as I keep saying, these species do co-occur. So I do understand that perhaps a finer resolution would have given us more clarity, but unfortunately, I just didn't have that data available. So I looked at the 7 species individually and over 30 environmental variables, including the traditional climate layers, as well as soil data. And from this, oh, sorry, I jumped ahead of myself there. Scratch that. First, I looked at the diversity of Triodanis across Texas across the United States using all that data I was just talking about. And as we kind of already knew and expected to see by these warmer colors representing where the concentration of species is. So all 7 occur in Texas, which was well known already and why most of my field work occurred in Texas. And then kind of as you go northeast, the species start to dissipate. Um, and the other really interesting thing about this map is that everywhere you see blue just represents Perfoliata. And then everywhere you see this green is going to be where Perfoliata and Biflora overlap, which, as you can see, is across most of its range. So I'm going to keep saying it. They really, really like to hang out together, but that lets us ask some really interesting questions. So now that other stuff I really wanted to talk about, by comparing their niches or the niche space. Like I was mentioning, this was at 1 kilometer, which is fairly coarse. So basically that means each square on my map was like 1 kilometer wide, which is pretty big space. And I found that relatively few species differed in niche space at this resolution. However, one that did come out as consistently different is Leptocarpa over here. And I tried to compare a Leptocarpa habitat to like a traditional Perfoliata for most of the other species. And you can see they are kind of different. Leptocarpa is generally more rocky and arid, while the remaining species grow kind of in more disturbed areas. Usually they're just like along the sidewalk and usually in damper spaces. And all of this, I had a lot of help from Morgan here, which we had a blast because we got to go then present all this data at the BSA conference in Alaska, which was also very fun. So, all of this work that I've talked about so far, as well as other work that's being done in the lab, especially by a lady who is currently working on chromosome counts for as many of the species as possible, will be compiled into a one genus-wide taxonomic treatment, which basically goes through everything we know about every species and catalogs it nicely as a description paper. So once I'm done with all this, I will really get started on getting that ready for publication. Okay, so that brings me to my final topic here, which is where I explored, sorry, population connectivity using paleoclimates. So I was broadly interested in biogeography and how individuals move across the landscape, and how they move over time, and then what factors influence this movement. So, as I'm sure many of us know or can kind of assume, remnants of what happened in historical climates and populations is going to impact the patterns we see today, because what we're seeing today is the result of evolution that happened previously. So to kind of do this, I built off of a previous student's work, and she used just the one most widespread species, which is Perfoliata. And they had rad-seq data representing these populations you see here. And she used this as well as climate data to explore the role contemporary factors had on population connectivity. So Morgan first did structure, which is this colorful bars you see at the bottom here, to look at the genetic relationship between these populations. She then, if we go up here, looked at the diversity based on breeding system at each population, and how that related to, or how breeding system variation related to diversity across these populations. And then for B, C, and D here, she was looking at different relationships based on distance and climate data. And generally across the board, she found that distance is a good predictor of genetic connectivity, which logically this makes sense. The closer together populations are, the more likely it is that there'll be some sort of gene flow than populations on the opposite side of the map. So to expand on this, I wanted to add paleoclimate, which ranged from approximately 8,000 to 3 million years ago. So I use this same RADSeq data as well as 6 paleoclimates. And my goal here was to map population connectivity through time. So as you can see by our little key here, these redder, more warm areas indicate areas of high connectivity, and then blue would be low. So my final model here said that the early Holocene and the marine isotope stage were kind of the most significant in explaining the patterns that we are seeing in this contemporary genetic data. And these two are kind of on both ends of the time spectrum. So about 8,000 years ago, and then about 3 million years ago. And this really highlights the significance of refugia and climate stability as important in defining the contemporary patterns we would see of population connectivity, which again is kind of what you would expect. The more an area kind of stays safe for an individual, the more there will be the ability for gene flow there. And and just a little humble brag, we did already publish this, and Brianne did a lot to help me with this as well. So if you take all my research together, I was able to add some well-supported and much needed phylogenetic context to the genus. And I was also able to provide a comprehensive study across phenology, morphology, ecological and climatic variables, and really for full like interdisciplinary approach to elucidate the evolutionary and ecological relationships within Triodanis. And that brings me to my acknowledgements here. A big shout out to both the Weber and Brown Lab. I actually could not have done this alone. That would have been crazy. And I know Kimberly is on Zoom here, and you can see her here. She was so nice and helpful. We met on our naturalist actually, and she just really liked Triodanis. I don't know why. But she was nice enough to actually go out in the field with us one season and come back and try to figure out this really cryptic species with us. So that was incredible. And with that, I will take any questions.